Hello everyone, welcome to my channel Bani's Biology. Today we will learn about the overview of the DNA repair mechanism. So life runs on a simple principle, that is DNA makes mRNA and mRNA makes protein. This is the central dogma of molecular biology, and this protein builds and regulates everything in the cell. But this flow of information depends on the integrity of the DNA sequence. Even a single error can miscode a protein, disrupt a pathway, or trigger disease. But what happened when the DNA, the master code, is damaged? So today we will explore how damage leads to mutation and how cell fight back with sophisticated repair system. First we will learn about how the DNA is damaged. DNA is damaged in two different ways. One is exogenous sources, another is endogenous sources. The exogenous sources that arise from the environment, and that's from outside the cell. That is ultraviolet radiation, ionizing radiation, and chemical mutation like tobacco smoke, industrial pollutants, and alkylating agent and viruses that affect in the DNA and also damage the DNA. And endogenous sources that arise from normal cellular metabolism, that is the byproduct of cellular metabolism, that is radioactive oxygen species, spontaneous hydrolysis and replication error, and alkylating agent that damage the DNA. For the DNA damage, there are different diseases happened. One is cell cycle arrest or senescence. That is a temporary or permanent halt to prevent division of damaged cell. And another is apoptosis, that is called programmed cell death, where remove damaged, aged or unnecessary cell in the control and beneficiary way. And another is cancer development, that is DNA damage lead to uncontrolled cell division and genomic instability. Genomic instability that refers to an increased tendency for mutation, chromosomal rearrangement, or structural abnormalities to occur in cell genome that increases the cancer development and also others diseases. So when damage isn't repaired, it becomes a mutation. There are different types of mutation. This is the DNA and this is the normal gene. When the normal gene transcribed and translated, then produced a normal protein gene product, and that developed a normal phenotype. And when this normal gene is mutated, that is mutated gene, that translated a abnormal, that is non-functional or no protein gene product, that developed a altered phenotype. So there are different type of mutation. One is point mutation, where the single basis is changes, that is G to A. Another is insertion or deletion, where the basis is inserted or basis is deleted, that causes phase shift. And chromosomal rearrangement, that is translocation and inversion. And also mutation can be silent, no change in protein, and missense that called altered amino acid, and nonsense that create premature stop codon. And also happen loss of function or gain of function that affect in gene activity. Some somatic cell mutation can lead to cancer or aging. So to prevent mutation, cell deploy repair system. Cell use multiple DNA repair pathway to fix different type of damage and maintain genome integrity. These include direct reversal, single stranded repair, and double stranded repair. In direct reversal, where the reverse is damaged without replacing bases. One is photoreactivation, another is alkylation. And single stranded repair system, where the one DNA strand is damaged, but the another strand can guide repair. And double stranded repair system, where the both strand is damaged. In single stranded repair system, there are present base excision repair, that is called BER, that fixes small lesion like oxidized bases. In base excision repair, uses DNA glycosylase, epiendonuclease, DNA polymerase and ligase. And nucleotide excision repair, that is NER, that remove bulky distortion like thymine dimers, and that first recognized by the damaged that present in DNA, then unwound and dual insertion and gap filling. And then is mismatch repair, that is MMR, that correct replication error. In this repair system uses mute-S, mute-L homologous endonuclease, polymerase and then ligase. In double-stranded repair system, there are present non-homologous enjoining, homologous recombination, and microhomology mediated enjoining. If the DNA is damaged or mutated, then DNA repair system will work. But if the any defect in the DNA repair pathway, then some disease will develop. One is Xeroderma pigmentosum, that is extreme UV sensitivity that causes skin cancer, and that is developed by the defect in the nucleotide excision repair system. Another is Lynch syndrome that causes the colorectal cancer, and that is developed by the defect in mismatch repair system. And another is BRCA1-2 mutation that causes breast and ovarian cancer, and that is developed by the defect in the homologous recombination repair system.